Yeah, so today I'm thrilled to welcome Dr. Andrew Satterlee, a brain cancer survivor and a dedicated scientist who's changing the way we develop and test cancer therapies. I had the honor of meeting Dr. Satterlee at the National Pediatric Cancer Foundation's annual research summit, and I swooped him up and said, "Can you be on our podcast?" So Dr. Satterlee is an assistant professor at the University of North Carolina at Chapel Hill and the director of Screening Live Cancer Explants Program and core facility at Eshelman Innovation, an academic institute focused on translating groundbreaking ideas into real solutions for patients. Since beginning his PhD in 2011 as a National Science Foundation Graduate Research Fellow, Dr. Satterlee has spent over 14 years developing new cancer therapies and better, more predictive models to test those therapies. Through his work, he's seen firsthand the limitations of traditional cancer models, from the artificiality of assays to the high cost of animal studies and the overwhelming lack of standardization across the field. This work is deeply personal for him because at just 20 years old, he was diagnosed with a rare heterogeneous brain tumor. With no clear consensus among his doctors, it was ultimately up to him, the patient, to choose between standard of care whole brain radiation or a newer chemotherapy option with promising early data. It was in that moment that he knew there had to be a better way to help providers and patients make informed treatment decisions, and today he's building that tool. In 2016, Dr. Satterlee began developing a unique drug screening platform alongside an interdisciplinary team of scientists and clinicians, including pediatric neuro-oncologist Dr. David Cram. Their system allows for real-time testing of patient tumor tissue taken straight from the operating room to the lab. It's already being used to help identify more effective therapies, minimize reliance on animal models, guide clinical trial enrollment, and inform real-time treatment decisions for patients with brain tumors, as well as other cancers like ovarian and lung. Dr. Satterlee's work is bold, deeply needed, and deeply human, and I'm so honored that you're here with us today.
Wow. Thank you. Maybe you just end it right there, huh? Thank you so much.
That's the whole podcast.
Yeah.
Obviously, Kelly and I know firsthand how this can affect people. It can affect you, and you don't even know it's going to. And I think that's the crazier part, is people are just living their lives day to day, and they think, oh, that could never happen to me. That could never happen to me. And then you wake up one day and it is you. And so, thank you for all the work that you do for all of these patients who are going to be able to benefit from this technology that you're developing. So can you explain kind of how this technology works and how it's pivotal in advancing your research that you've got going on right now?
Thanks, Tiffany. It's a great question. We have a really strong partnership with the surgical suite and the doctors at UNC. And so we get the patient tissue right from the operating room sent to our lab. And we just put the drug right on the tumor. So instead of doing mutational assays to see what mutations your tumor might have and what drugs might work, or looking at the stained microscope slides to see what the tumor looks like, that all can kind of inform what treatment you should get or what drug might be a good one to develop. But it doesn't answer the direct question, which is the tumor going to die? So we just put the drug right on it. Our secret is we keep the tumor alive in our lab and then ask that question. And in just a few days, we have the answer whether the tumor lived or died in response to the treatment.
Wow. It's so interesting to me that this has not already been the standard of care, because listening to you talk about it, it makes so much sense. So what led you to start exploring the idea of testing cancer drugs on still living tumors?
Yeah, you make a good point, Kelly. It seems like this should be a simple, easy way to do things. And folks tried it a couple decades ago, and the technology just wasn't there, or the understanding about tumor biology to really get out meaningful answers. So We started this a while ago because we were testing out a cell therapy in the lab I was a postdoc in. And we didn't have, we felt like, the right tumor models to ask the questions we wanted to ask. So we started trying to build, heterogeneous tumor models that had some of this cell line and some of that cell line. And we realized even this isn't good enough. We need the real thing. We need the real patient's tumor. But this is a really hard thing to get. It's hard to go into the clinic and just find somebody to give you some tumor or figure out the right way so you do it with the right regulatory standards and you're not doing something out of bounds. Because we need to be careful about that too and respect this really, really special gift that we get from these patients. So it was a long road to figure out how we do that, how we get the tissue into our lab, how we keep it alive, how we test the samples. And working with Dr. David Cram, who you mentioned, who's a big person in the NPCF world, has been a really, really great help for us.
This is like so cool. I can't even imagine like being able to tell a patient whenever they get out of a surgery, hey, this is the drug that's going to work for you. That's so cool. So what was your initial reaction whenever you very first tested somebody's tumor, like outside of their body? Like, what did that change for you?
I remember this. So it was like February 2020. So we had to take a little hiatus after that. We got some great data. The very first tumor we brought back, I was still on the bench then. So I went to the tissue procurement facility that we have at UNC that helps with this, brought the tumor back, put it out in the cell culture hood. I had my gloves on and my coat on and everything, and was looking at a patient's real tumor. And at that moment, okay, now what do I do with this thing? We were kind of making things up as we went. We had an idea about what would work and what wouldn't work. And then the end of our assay is only 4 days long. So at the end of that time frame, oh my gosh, this tumor is still alive. We could test it with drugs. We could do all kinds of things. And then the world ended for a while in March. And right after we'd gotten a second tumor tissue, and a lot of things changed, but it actually helped us. A lot of consent forms that patients got moved to e-consent, so electronic consents, and things like necessarily changed, but then have remained and got easier for us to get more and more of that patient tissue. But it was a real eureka moment for us.
Absolutely. I love that you remember the exact date.
I remember you.
It does stand out too with everything that was going on in the world.
Yeah.
So what challenges did you face in getting this technology to work with living tumor tissue, and how did you overcome them? You mentioned that this was done, tried decades ago and the technology wasn't there. So tell us a little bit about some of those challenges.
One of them is that every tumor is different. So this is a part of the great thing about what we're doing and the difficult thing. Cell lines and mouse models, you can count on them to work every time because it's the same thing every time. The patient tumors are tough to treat in the clinic because every tumor is different. This is, if you ignore that part of the equation, your treatment's not going to work on everybody. You're not going to be able to get the right answer. So understanding that every tumor is different, and some, when you get them into the lab, you're going to be able to, mince it up really well and put the right kind of reporter stuff on it, and it's going to survive really well. And others, you're going to get it in the lab and it's going to be a sticky mess. I don't want to get too in the details about what these tumors look like, but your real patient tissue is coming into our lab. And every one of them presents its own unique challenges. But that's how we have to be treating the patient as well. So we got to figure out how to make a process that works for everybody, not just the high-grade gliomas that love living anywhere because they're so aggressive, But the low-grade tumors, the low-grade gliomas that are very, very common in kids, metastatic tumors, there's a lot of different types of tumors that aren't like the splashiest type that everyone wants to talk about, that a lot of people still get. And we have to know how to deal with those also.
Absolutely.
I think that we harp on that a lot about tumor types being so different. Each cancer type's different. Glioma is different. They all respond differently. And I think that you're going to be able to answer one of the biggest questions in oncology treatment is, why does this drug work for this tumor and not for that one? And I think that that's absolutely incredible.
That's what we're aiming for. That's what we want.
Yeah. So, like, how does the testing that you're doing compare to being tested on mouse models? I mean, I think we've kind of answered this a little bit, but dig into that a little bit more.
Yeah, it is. So cell lines in a plastic dish are kind of on one end of the spectrum and usually in a lab. And mouse models, where you put the tumor literally like into the head of mice, it's on the other end. And the cell lines are so artificial, but fast. You can get the answer really fast and do a lot of things really fast. The mouse models are expensive and they take a long time and pretty opaque, right? You can't really see what's going on until the end of the study. Sometimes you can see if the tumor's growing or not. And you use a lot of live animals to do this. And we think that the way that we do the assay with the patient tumors is kind of the best of both worlds. We get the real patient tissue, but we can do the assay really fast. And so we think when we're talking about a drug development pipeline, testing out a new drug is going to be easier, faster, better, more relevant results with our system. But when we go down the road toward actual patient treatment guidance or clinical trial enrollment guidance, we think we have a better model for that as well.
Yeah, so tell me more about that. You mentioned that this approach could lead to more personalized treatments. So how do you see this technology kind of transforming the future of cancer therapy?
Yeah, I think, and I'll just repeat kind of those three buckets that I think this could really help in, is new drug development. You've got a new idea for a drug or a few different similar drugs. Which one's the right one to bring forward? We can help with that by not just treating cell lines or mouse models, but the real patient tumor. Does it work on the real thing? Second, if as this drug moves down the development pipeline, you're going to want to try it in real people and enroll those patients onto your trial. Right now, decisions for who goes into a trial and who doesn't get to go in that trial are based on, do you have a certain mutation? Do you have like the BRAF V600E mutation? You got to have this to get the BRAF inhibitor that's hot off the press, right? The new drug. You got to have that right mutation. Well, what if the cutoff point was your tumor dies when we give it this drug in the laboratory? Then you get to go on the trial. And if we get the tumor into our system and we put the drug on it and it doesn't die, then don't join the trial. You're not a good candidate. Instead of doing the microscope staining to make that decision, just put the drug right on the tumor. So that's kind of the second bucket. And the third one is, can we actually guide treatment of you, not on a trial, but can we give the results to the doctor, to you, the patient, and help guide what drugs may work for you in the clinic. Now, we're not to that point yet. This is what we're attaining to, but it's exciting to see ourselves go down this road.
So
Absolutely.
exciting.
It's exciting for me too.
Yeah, that's what I'm thinking myself. I'm like, this is so cool. So, I mean, you focus on brain cancer. We heard in your bio that you have had cancer yourself. So can you tell us a little bit about your personal experience with cancer?
Yeah, thanks. This is it's like a dream job right now, and I'm kind of each day can't believe that I get to work on this, because in 2007 I did have a brain tumor. had hydrocephalus, which means that the tumor was growing in a part of my brain that didn't allow the fluid around my brain to drain. So it was building up. So I got, headaches worse and worse, nausea worse and worse, and went to the hospital and they did a CT scan and found the tumor sitting there. And then there was a long saga that you both are very familiar with after that. Came home I got a shunt in my head that then ran down under my skin, down into my belly to drain the fluid that wasn't draining the right way. And then a couple of months later, got the tumor taken out at University of California, San Francisco by the best brain surgeon in the world, Dr. Mitch Berger, who I credit for a lot of things. But the day we came home from that initial diagnosis is when my parents got caller ID. And we didn't have that at that moment because there were going to be a lot of calls coming in, and it just changed a lot of things overnight. Like you said, Tiffany, you just one day, I'm in college, sophomore in college, doing the college thing, and the next day, a lot of things are different.
That's incredible.
Yeah, I had surgery to remove the tumor in December of 2007, and then I had chemotherapy and radiation therapy after that, and have been cancer-free now almost 18 years.
Wow.
Love it.
The fact that you've come full circle and been able to use that experience. Now, did you know at that point, because you said you were a sophomore, did you know that this is what you wanted to go into? Like, was that already in your, or did this kind of say, nope, this is what I'm studying now?
This was the big moment of, okay, now I'm going to do cancer therapy because we were doing research about what my tumor needed because there wasn't a consensus, right? Like you said, the doctors didn't agree what to do. We had to figure it out. I wrote like a research report for a class on what's the right drug for a heterogeneous germ cell tumor, because that's what we were trying to figure out. I was doing research on my own brain tumor. And, I've always kind of been a question asker, someone who wonders things. And so I think science was always in my future. But doing cancer research is what I gravitated to after that. And I did my grad school in a lab that wasn't focused on brain cancer, but it was some kind of cancer. And I thought, okay, this is really close to what I'm doing. And now, I did my postdoc and started working with all this stuff in a lab that did brain tumor research, brain tumor resection surgeries, giving mice the same procedures I got, the same chemotherapies I got. And I'm sitting there doing procedures like that, thinking, I am so lucky to be working here late at night and working extra hours to do all this stuff. It's like a dream come true, and it's really, really hard work, but I just am so blessed. I'm so happy that I get to have this as my career.
Well, we feel very lucky that you have this as your career, because having somebody who's so passionate about it and really cares and has a personal tie, that means the world to us cancer moms. So thank you for doing what you're doing.
To the point about, you call yourselves cancer moms, and I get that so much. I think my mom was the person most changed throughout my cancer journey. She, obviously always a wonderful person, but leaned into her faith so much and leaned into, has become so, I am so in awe of who she is and the way that she handled things going through, I would rather be me than her in the whole situation. If I, thinking back about how it all went down.
Yeah, no, it definitely is hard. It's hard to be the mom in this situation, but you got to give yourself some credit too, because what you went through is not an easy thing for anybody. So we talked about, becoming a scientist and how that was kind of always, I think, meant for you. But can you tell us a little bit about the difference between an academic scientist approach and a translational science approach, and why the latter is crucial for patient care?
This is a great question. I love that you're thinking about the difference of these two types. So I was in I interned in a basic cancer research lab, or not cancer, just basic research lab. And these labs are important. So, like, let's learn something new about the world as a basic academic researcher. This lab wanted to know what specific little molecule was responsible for a mouse knowing that the other mouse was a boy, right? So it's like, this is important. I think if you could find that out, whoa, like it fundamentally
Yeah.
changes your the concept of what like scent is and what maleness is, and like what, there's something like this. And they published right before I got there a paper in Nature, like a great huge paper. They were doing all this work. The guy came from a Nobel laureate's lab, and I asked him, so okay, like now, like if we find this out, what's going to happen? Like, what are you going to do with the information? And he said, we publish it, and it's now part of the literature the world gets to see, and we let someone else figure out what application that gets. And yes, that is so deeply needed to have basic research, learning new things all the time. But it's got to be balanced by the translational research that takes those things and does something with them. So that's what resonates with me the most, is that translational science, where we have applications from the research that we're doing that directly affect patients. And I think that there's a difference. One is not necessarily better than the other. They're both needed, but when you're focused on what patients need, how do we change patient care, how do we really do, you got to focus more on the translational side.
Absolutely.
So, in your opinion, like, what are the biggest hurdles in the path from the idea of this to the bedside when it comes to your research?
Say that one more time, Tiffany. I'm so sorry. Say that one more time, Tiffany.
Oh, sorry, y'all. We're having like the sound issues today are for real. But okay, so in your opinion, what are the biggest hurdles in the path from the bench where you're studying this to taking it to the bedside where we can actually use it?
Yeah. It is a tough road to find new therapies on the bench and get them to work in patients. We're trying to bridge that gap a little bit better, but we also want to make sure that the things that we make work and don't cause harm. And so there's a lot of regulatory work in there, and maybe there's the road is longer than it should be and more expensive than it needs to be. But cancer is smart and innovative and devious and really, really hard to treat. And the creative solutions that researchers are trying to come up with are so deeply needed, but also the most difficult to treat cancers are so, so, so tough. So we need to learn more about them with the basic research, and then build things that respond to those really tough traits. So there, part of the issue is, it's just a long slog to get things to patients from a regulatory standpoint and drug development standpoint, a funding standpoint. But part of it is cancer is horrible and a genius, and really just a word for cells that are more fit to grow and live by themselves and be greedy for all the resources in your body, but don't play well with the other cells in your body. So it's such a general term. Like, how do you defeat, greed? It's a really tough problem,
Right. Absolutely. And I love the way you explained and looked at it, that every tumor is different, because that's something Tiffany and I hear a lot. Like, how come the world we can do this and we can do that? Have you been seeing the posts about how come they can create the dire wolf, but they can't cure cancer? I've been seeing that everywhere. And it's hard for me to bite my tongue and not respond to all of these with cancer is not one disease. That's the thing. And I can't harp that enough. We've said that on like every podcast we've had. It is not one disease. It is every tumor is unique and different and therefore it has to be treated different. So you're not curing one thing.
Like
You're so right.
cancer
You're so right.
type that's the same is also different. Like
also-
it has different molecular makeup, and that's what makes your work so incredible is that you can see that in real time how different these tumors are, just like right there in
That's
front of your
right.
eyes.
David Cram is currently seeing two patients, two 3-year-old girls who both have a low-grade glioma with a BRAF V600E mutation, molecularly identical. They both got the same treatment that has a 50% chance of working. And one of them it worked on, and one of them it didn't work on. What's different about these tumors? The way that we're characterizing them from the mutational status, from how it looks like on the microscope, it's not getting the difference. But in our lab, when we put the drug right on the tumor, we're going to see if it lives or if it dies. We get to look at all the co-occurring mutations and complicated things that are going on at the same time that you don't get when you just pick out the one mutation you think is driving the whole thing. We're looking at everything together and how it works together. And then maybe we can get better at knowing who should get this drug.
Absolutely. Absolutely. That's just incredible. I'm so excited about this science. So, all right, I came up with this question at random because I actually read an article where you kind of talked about this, and I wanted to hear your thoughts more on that. You mentioned that what journals and what grants want is different about what a patient needs. And I found that really interesting, and I thought, I'd love to hear his thoughts on that some more.
Yeah, I got this kind of disillusionment in my head while I was in grad school a long time ago, and I still feel like this is true. The journals and the grants really love splashy new things. And you need to have something splashy and new to get your new grant, to get a paper in a high impact journal. But the patient needs something good that's made a little bit better, innovation on top of this iterative process. And it hit home for me back in 2015 or whenever it was, when I was in a lab that made nanoparticles to deliver, like specifically deliver drugs somewhere. And a grant reviewer criticized my PI's grant because he said, okay, like you already made that nanoparticle, a few years ago. You already got a grant about that. We're excited to see the next type of nanoparticle you make, the next thing that's different from the one you have now. That's what we're going to fund you for. And he shot back and said, I like the nanoparticle I already have. And we think we can make it better, a little bit better and a little bit better, and make different applications for it over time. If we start back at square one with a new, different thing that you are going to give money just because it's new and different, we're not giving the patient what they need. That's what really, there was a switch that kind of flipped then. We don't need the new splashy thing every single time. Let's work on building this thing that, I've been building this thing for 8 years. There's been people who've been building their technologies or their drugs for 15, 20 years, a lot more. Like, can we innovate on the best things we already have instead of making something that looks really cool but could never get into a real patient because it's so complicated or so expensive or just behind the curve now that you have to start all over again?
I feel like that's a really interesting topic, and those are really amazing points that, hopefully we can bring some awareness with this.
Yeah.
It's the new splashy thing is cool, but it's, sometimes it's the nitty gritty stuff that you're doing in the lab that might be cool to the three of us, and maybe not other people.
Yeah, that's right. That's right.
Our science-minded people are like, no, that is cool.
Absolutely.
Yeah. You don't always need to make something brand new.
Right.
But that's what innovation sometimes wants to look like, or you get the most clicks.
Yeah. So with the opening of the core facility for the patient tumor testing at UNC, how do you envision the collaboration with other researchers to accelerate drug discovery?
Yeah, we opened a core facility. This is actually, I think, it's become a pretty cool thing for us to help us expand our reach. We have been getting patient tissues and testing them and like building a platform to do the testing. But I don't have a novel drug. I'm not. My career is not going to be about finding the next drug or develop. Like I'm not a chemist. I'm not a drug developer. So we want people with drugs to collaborate with us because we've got a better way to test those drugs. And so we opened the core facility, been working with groups inside UNC and even groups outside UNC, academic and non-academic groups. And we just a couple weeks ago published a paper with Dr. Aziz Sanjar at UNC, who won the Nobel Prize a few years ago at UNC, has a new drug that he wants to get toward people. And he used our core facility and tested it on our patient tissue. Now we've got a paper out, and he's got new data that we're working with together that is unbelievable. This first paper is really, really cool collaboration. The new data that's not published yet is looking really good, and we're going to see how to move his drug down the field. Similarly, there's other groups that have tumors that they want tested, right? Hospitals or clinicians that say, I'm taking tumors out all the time, but I'm throwing them away, or they're going to pathology and then, just doing what they need to do for the standard. Like, how do we test these in a better way? So we want to be able to accept tumors from other sites. And we're getting really close to doing that at University of Florida, at University of, at a couple other places. It's really exciting that this has kind of opened up a real bona fide route to collaborate in these interesting ways.
That's incredible. You should be very proud. That's a huge accomplishment.
Yeah, that makes
So,
sense.
as someone who's deeply involved in both science and clinical aspects of this cancer research, what advice would you give to, let's say, a young researcher who wants to make a big impact in cancer treatment?
I like the idea of wondering things. If you're it depends on how young you are, but like a young person, like a middle school, high school person, I think just keep wondering things, keep asking questions. I kind of, to a fault, ask questions like at restaurants, at the hotels, to my wife. You just wondering things like, oh my gosh, would you rather questions, just random stuff. But this is who I am, and I just can't help it. So I do it with my career also. But also to the slightly older researcher who might be in grad school or struggling over the day to day of getting data and hoping your results are good. Everybody who does research falls into the pit of despair. It happens. There is so much failure. It's unbelievable how you have to get tuned to constant failure, and there's something about building confidence in your process, right? That you design the experiment right. If you're confident that you're designing the experiment right, that's all you can do. Maybe your hypothesis is not supported, right? You made a good guess. You tried the thing. The world doesn't work that way, and now you know that. As long as your process is sound, you will be able to put one foot in front of the other and eventually get out of the pit. And if you're asking good questions and your process is good, you'll eventually make it through this iterative, process-oriented position. But it's a slog. That's what research is, and it's just a tough thing. So I think having confidence in that about doing the process right is important to have in mind.
Good advice. Keep asking
Yeah.
the questions.
Yeah. So, I mean, you're talking about how people can get into research, what they need to do. And obviously you said that there are failures, there's hurdles to overcome, but what's been the most rewarding moment of your career thus far?
Man, it's a really good question. There's one moment that really sticks out, and it's all kind of tied with my connection as a patient myself. But uh, I've kept in contact with Dr. Berger, who took the tumor out of my own head. And in 2016, I met him at a conference. First time I'd met him like as a peer. We shook hands and had the same, lanyard on at the Society for Neuro-Oncology conference. And he was like, winning some award or like moderating the whole deal, whatever he did over there. And I shook his hand, and he turned me around and he said, "Let me see that scar." And he put his hand on, on the scar on the back of my head. And we talked for a while, and we've been, keeping in touch in different ways. But that realization that I'm on the other side now, I'm working with this guy, uh, and in a field sense, that was so significant. And that was 9 years ago. So I've been doing this for a long time since then. But oh my gosh, I still think about these interactions that I get to have with him and how special that is.
It's awesome. I feel that way when I go to our research summits, to be honest, being on both sides of it, but as a cancer parent and being in that room and listening to the way you guys talk on stage, the passion that you have for what you're doing in the lab. Those are the moments for me. So I can relate to you there, and that's really awesome.
Yeah. I mean, even for me, who, my daughter obviously doesn't benefit from anything that we're doing or that you guys are doing, but it helps benefit that parent or that child who is going to hear, "Hey, you've got cancer." And for me, that means a lot, because I don't want another parent to have to hear the same words that I heard. So it's so special, and full circle moments all around.
Absolutely. Speaking of that emotional work that we deal with, cancer research is obviously intensely emotional to work in. And being somebody who had to deal with it personally, how do you manage the emotional side of dealing with something as devastating as brain cancer and still focusing on the science?
Yeah, there's no way I could be a physician in this field. I thought about med school for a while and becoming a doctor, and I don't understand how David Cram does it or how any of these people can. It's a special position that they're in to help kids in that way. I stand a little bit farther from that, but there is a balance, right? If you don't focus on the patient enough, you get mired in your science. And if you focus on your science too much, you just get lost in the data and getting your statistical p-value to be what you want, and your paper published and your grant money in, keeping the lights on in your lab. It is so easy to just keep your head down all the time, because it's hard to keep the lights on in a laboratory. It's just really, really tough, especially it's just really hard to get good data constantly and good ideas constantly. But you can't only focus on that, because then you're missing the point, which is the patient. You've got to make sure your stuff is translatable, that you can get it over to the other side, to the clinic. And if you're not focused on that, I had a person who said, "I am so successful." He was retiring. "I've been so successful in my career. My H score is so high," which is like the amount of papers you have with a bunch of citations. "But my therapies have not helped a patient yet." And he's thinking about his legacy. And of course he's done a huge amount of work for the field. A huge, like, his, the basic research that he's done and like toward applied stuff. Of course his legacy is impeccable, but he felt it in himself that he didn't do what he really wanted to do. That has stuck with me.
Yeah, being able to take this science and all these hours you're spending and actually have it work and fix a patient and save lives. But, I just want you to keep in mind, and for all the scientists we've interviewed and talked with, yes, you're in the lab and yes, you're doing the nitty gritty stuff, but that's where it starts. And that is, so extremely important. Without you doing the research you're doing, there would be a lot of kids who wouldn't have a future. And so what you're doing is just extremely impactful. So what's next for your research? How do you envision the development of this within the next 5 to 10 years?
Yeah. Oh my gosh, what a question. Ideally, we'd be working with a lot of people who have new drugs they want to move forward, and we'd be working with a lot of folks who have tumors that they want characterized in this way. And I think that the next real concrete step for us is to be embedding our technology in other people's clinical trials. At first, just as to correlate with their results. So if the patient on their trial can get a specific drug, and then that tumor can come out of the patient's head and get that same drug on our system, we can then really start more quickly getting correlative results to say, are we really predictive? We need more and more and more of that data. Because right now, we're relying on a lot of tumors that just we get out in the wild, right? The patient's glioblastoma comes out and comes to us like an emergent surgery, comes to us, and that patient gets who knows how many different therapies at different times. It's messy. And so it's hard for us to know, all right, we're going to give the standard of care drugs and some other things, but this patient's getting so much stuff and it's changing all the time. How do you know if you're predictive or not? It's tough. So we're slowly getting data that's making sense and looking like we're getting a lot of really cool new data. But over the next few years, we want to be really systematic about this and partner with folks who want also, I think it can be a win-win where we are helping understand our own system if it's predictive. And then if it is, then that therapy can continue to use our system to guide the development of their drug.
How long would it take? You're putting these medications straight to the tumor slice, the brain slice itself. How long is that process potentially?
Yeah, just a few days. So the tumor comes out of the person's head. We have a cryopreservation protocol we've been using. Started out because sometimes we get tumors so late at night, and then it would take so long to process everything. You'd just be there all night long. So we thought, what if we could just freeze these down and then wake them up later? But now we can freeze down several different vials of the same person's tumor. Like we mince it up, and we, several representative bottles of this we keep frozen, so we can wake one up and ask a quick test with a few drugs, and then based on that, we can wake up another vial. So we don't have to ask all the questions right away. So then to that point, if that's our standard protocol, we get the tumor, say midday on a Wednesday, we freeze it down, we wake it up the next day on a Thursday. That's like the earliest. We wake it up, then one vial up the next day. Then we, by the next Monday, 4 days after that, we have the answer, whether it lived or died with the drug treatment. So all in all, that's a Wednesday surgery and a Monday result.
That's incredible.
Yeah. I mean, that's like better than any other tech, any sequencing, anything that we can do right now. It's so cool. Oh, no, go ahead.
We do like the fast assay. People ask us often, how long can you keep the tumor alive? But we have never really asked that question. It's how fast can we do it?
Love it. So what would you change about the current cancer research landscape, and why would you want to change it?
Oh, man. I think, I think a lot of groups are siloed and don't talk to each other as much as they can. And I think that there's connections between the research lab and the hospital, like clinicians and surgeons, that I feel really lucky that we've been able to build these good relationships with David Cram and Dominique Higgins and different folks over in the hospital. But that's more rare than it should be. But even more than that, connections between our stuff at UNC and then what people are doing in other laboratories at other universities. It's so important to know what other people are doing and work together collaboratively. But that's also tough because you got to keep the lights on in your own lab, and you've got to be an independent scientist that has your own thing you want to move forward. So if we're going to collectively choose, one or a few good things to move forward, then there's winners and losers, and that's not good either. So I don't know how to solve that other than let's just talk to each other a little bit more.
Absolutely. I think you brought up amazing points today about, some of the hurdles that we face and why things take the amount of time they do. Some of the hurdles between practicing in the lab versus applying it to patients. You've told us about this incredible technology that I can't wait to see where it goes here in the next few years. So, we really, really appreciate you today. Thank you so, so much for joining us.
Oh, this was absolutely my pleasure. I had a great time. Thank you.
So how can people follow you or find you or look into your research?
Oh, that's a great question. I'm so bad at social media, so I need to be better at this. We have a website. We have a our core facility has a website. Our lab has a website. It's if you look up screening live cancer explants or just my last name at UNC. Actually, I think we are actually slice.unc.edu is the acronym for screening live cancer explants. If you look that up, then that's
nice.
A website. Yeah.
I love that play. Yeah. I love that play on your research.
Funny acronym. There's a few different ways, but please reach out. My email is almost my last name, but SATTERLE at email.unc.edu. Please reach out, send me an email if you're interested in collaborating, or send a message. I think that's the right way to do it. There's
Perfect.
on the website.
We can link
there's
all
ways
that.
to contact us. Yeah.
Yeah, we'll link all that in the show notes today too. So if anybody wants to email you or collaborate, I think that would be amazing. I think that you're incredible. This research that you're doing is so awesome. Your personal story just makes it all the better. And we are grateful that you joined us today. And if you want to look into, for our listeners, if you want to help, donate, get involved, you can look in our show notes. You can go to goldribbon-kids.org or nationalpcf.org and get involved with anything that we're doing. Dr. Satterlee, we're so, so thrilled to have you today, and thank you for being with us. Join us next time for another episode of An Unexpected Bite.